Curing a thick slab of salmon isn't magic, it's chemistry and microbiology working together. Let's break down why salt and sugar are essential and what other factors you need to nail a perfect cure. Why salt? Salt is the primary preservative. By drawing water out of the salmon's flesh through osmosis, it lowers the available free water activity that bacteria need to grow. Question: what happens if you undersalt? You risk uneven curing and even bacterial spoilage in the center. Oversalt, and the fish tastes inedibly briny and dries out. As a rule of thumb, a 5–10% salt by weight cure 5100 g salt per kg fish balances preservation with flavor when you're working with a 2–3 cm thick fillet. Why sugar? Sugar isn't just for sweetness, it modulates texture and taste. It counteracts salt's harsh edge, creating a more rounded flavor profile. On the molecular level, sugar also contributes to water retention, so the salmon stays supple rather than chalky. Sugar can feed lactic acid bacteria in fermented cures like Gravlax, gently acidifying the surface and adding tang. Typical ratios run about 2–4% sugar by weight relative to fish. So what else goes into the cure? Nitrites or nitrates. A small amount of curing salt, Prague powder, one at 0.25% of fish weight, prevents botulinum growth and locks in that classic salmon pink color. Emulsifiers, vinegar or citrus juice, improving. In Gravlax, a dash of acidity helps denature proteins on the surface, improving a slight brightness to the cure. Aromatics and herbs, dill, juniper berries, citrus zest, peppercorns. These don't just flavor the fish. Some, like juniper, have mild antimicrobial properties that complement the salt. Temperature and time. Curing happens best at 2 to 5 DC. For a 3 cm thick piece, you'll need 24 to 48 hours, flipping midway to ensure even diffusion. Thicker cuts stretch to 72 hours, but beware of over-curing. The outer layers become too firm if left too long. How does the science play out? Diffusion kinetics. Salt and sugar migrate inward at roughly 1 ml per hour under refrigeration. Protein modification. Salt denatures surface proteins, firming the flesh. Sugar helps buffer that firming so the texture stays silky. Effective curing drops AW below -0.92, the threshold most spoilage bacteria need. pH control. In acid-assisted cures, lowering pH to around 5.5 further inhibits microbial growth and adds that signature tang. Example, quick Gravlax cure. 1 kg salmon fillet, skin on 60 g kosher salt, 630 g granulated sugar, 3%. 5g pink curing salt, 1.5. Zest of 1 lemon, 1 teaspoon paw crushed black pepper, a handful of fresh dill. Mix salts and sugar with aromatics, coat the fish evenly, wrap tightly in plastic and weight it down. Refrigerate at 3 DEC, flip after 24 hours, then check at 36–48 hours. When the flesh feels firm but still yielding under your finger, rinse off the cure, pat dry, slice thin, and enjoy. By understanding the roles of salt, sugar, nitrites, acidity, and time, you can adjust any cure recipe for thicker or thinner cuts, bolder or subtler flavors, and absolute safety. That's the science behind curing salmon. Delicious, preserved, and oh so satisfying. Curing is a preservation method that uses salt, sugar, and sometimes other compounds to draw moisture out of fish, inhibit microbial growth, and transform texture and flavor. The process works through osmotic dehydration. Water moves out of the fish into the surrounding cure due to a concentration gradient. Simultaneously, salt and sugar diffuse inward, altering the internal environment of the tissue. This reduces water activity, PY, the key variable that controls microbial life. The result is a semi-dehydrated, denatured protein matrix that is safe and palatable. Gravlax is traditionally made by rubbing a thick salmon fillet with a mix of salt, sugar, and spices, then curing it under weight for 2-3 days. It's like shrink wrapping the fish from the inside out. Moisture leaves and a protective layer forms within. Salt is the cornerstone of curing because it draws water out of the fish via osmosis and disrupts microbial cell membranes. High salt concentrations dehydrate bacteria, denature their proteins, and collapse their metabolic processes. Salt also initiates protein unfolding within the salmon itself, changing its texture to a firmer, more sliceable state. Additionally, sodium ions compete with microbial nutrients, lowering the risk of spoilage or pathogen growth. Without salt, curing would be unsafe and microbiologically unstable. A salmon fillet cured with 3-5% salt content becomes firm and resistant to listeria and botulism under refrigeration. It's like turning the fish into a biochemical desert, hostile to microbes, but safe and delicious for humans. Sugar balances salt's harshness, adds subtle sweetness, and contributes to preservation by also reducing water activity. Unlike salt, sugar doesn't kill microbes directly, but limits their ability to reproduce by binding available moisture. Sugar molecules also help retain some internal water, preventing the fish from becoming too dry or chewy. In addition, sugar can participate in mild Maillard reactions during further preparation or aging, enhancing flavor. Different sugars, e.g. brown maple or molasses, contribute distinctive profiles. Traditional Gravlax uses a 2-to-1 salt-to-sugar ratio to balance firmness with delicate sweetness. Sugar in curing is like a velvet glove over salt's iron hand. It softens the experience while still getting the job done. Thicker salmon fillets cure more slowly because salt and sugar need time to diffuse into the center of the flesh. The rate of diffusion is governed by fixed laws of diffusion, meaning time and temperature must be adjusted for thickness. If undercured, the core may remain raw and unsafe. If overcured, the exterior may become too firm or salty. Therefore, uniform thickness and proper timing, often 24-72 hours, are crucial for consistent results. Some techniques use weights or vacuum bags to improve penetration. A 2.5 cm thick piece of salmon may take 48 hours to cure evenly at 4 DC, while a 1 cm slice may only need 24 hours. It's like marinating a thick steak. Flavors take longer to reach the center, so time must match depth. Spices, e.g. pepper, dill, coriander, citrus zest, alcohol like aquavit or gin, and smoke flavor can be added to enhance sensory qualities. Some recipes also include nitrates/nitrites for color stabilization and added antimicrobial protection, although these are less common in home-cured salmon. Temperature control is essential. Refrigeration, typically 1 to 4 DGC/microbial growth, while still allowing curing to proceed. Weight or pressure can be applied to improve moisture expulsion and firm texture. Vacuum sealing accelerates curing and minimizes oxidative flavor changes. Scandinavian Gravlax is traditionally made with salt, sugar, dill, and aquavit, then wrapped and pressed with a brick or weighted tray. Think of curing like a culinary equation. Salt and sugar are the constants, but spices and technique are the variables that define the outcome. Water activity is the measure of free unbound water in food, and it directly determines whether microbes can survive and grow. Lowering water activity below tilde 0.9 is enough to inhibit most bacteria. Below tilde 0.85 suppresses even salt-tolerant ones. Salt and sugar both reduce water activity, but in different ways. Salt disrupts ionic balance, while sugar ties up water through hydrogen bond. The key to safe curing is achieving a final product with sufficiently low ODO without making it inevitably dry. Measuring or estimating AW is critical for commercial and scientific validation. Properly cured salmon has a water activity around 0.94, 9.96, safe under refrigeration but still moist and sliceable. Water activity is like the crowd size at a party. If too many free agents are around, bacteria join the dance. Salt and sugar trigger protein denaturation, the unfolding and reorganization of muscle fibers, which leads to a firm, sliceable texture. Water loss from the muscle allows myofibrillar proteins to rebind more tightly, forming a denser gel-like matrix. This is distinct from heat-induced denaturation, like in cooking, which coagulates proteins. Curing preserves a raw-like translucency while altering structure. The result is a firmer texture with slightly reduced elasticity. Cured proteins can also bind more flavor compounds, enhancing taste. The silky yet firm texture of lox or Gravlax comes from salt-driven protein unfolding and slight moisture loss. It's like drying a sponge just enough to make it springy and dense, not brittle, not wet, but perfectly flexible. Curing must be done under clean conditions with properly stored fish, as raw seafood can harbor dangerous pathogens like Listeria monocytogenes. Only sushi-grade or frozen and thawed fish should be used to avoid parasites. Precise curing times, salt concentrations, and refrigeration are critical for safety. Vacuum sealing and pH control can add safety margins, but are not substitutes for proper salt. For thicker cuts, slicing and examining the interior is necessary to ensure even penetration. FDA guidelines require freezing at -20 DEGC for 7 days or 35 DEG for 15 hours to kill parasites before raw consumption. It's like hiking a mountain with the right gear. If you miss a step, the risk isn't culinary, it's biological. Curing thick salmon fillets is a scientific interplay of osmosis, diffusion, water activity reduction, and protein restructuring. Salt removes water and preserves. Sugar balances flavor and texture. Thickness determines time, and environment controls safety. When done properly, curing creates a stable, flavorful, and safe product that's both artisanal and biochemically elegant. The process preserves more than food. It captures flavor through precision. In curing, chemistry becomes cuisine. Gravlax, made with 3% salt, 1.5% sugar, dill, and 48 hours of curing under weight, becomes a Nordic delicacy. It's like programming a biological algorithm. The right input and timing yield a perfect edible output.